tonight by talking a little bit about memory and learning. Several of you have asked me about study practices and things that you can do to help improve your scores. And a project I'm working on has me doing some research on this very topic. And I just thought I'd share with you some of the things that I've learned. First off, we're familiar with short-term memory and long-term memory. And short-term memory is just that, very short, 15 to 30 seconds. It's the memory you have while you're writing down your notes, trying to remember what I just said, and listening to the stuff that I'm currently saying, and trying to remember that it doesn't last very long. And you're soon saying, what did he just say? Or what was that? Okay, so short-term memory, very short, and you don't recall much of it. Long-term memory happens in two ways. It requires some protein modifications to take place in the neurons themselves, and then for extended long-term memory, you have to get protein synthesis to occur in those cells. The only way that happens is through repetition. So when we sit down in class tonight and I give a lecture, you'll hear me say something several times. I may antigenic variation or something like that. And you'll remember that term maybe for an hour to 3 hours because you've heard it in repetition several times this evening. That's because you've essentially activated proteins that are already present in those neurons for that type of long-term memory. But if you want to retain that memory for any period of time, you actually have to get protein synthesis started. And that's where additional repetition comes in. So after you've heard it initially tonight, you have to expose yourself to it again, and not 2 weeks down the road, but in a reasonable amount of time. And you have to do that several times over the next couple of days, but you have to provide yourself breaks in between study sessions. Turns out if you don't put those breaks in between study sessions, your cells don't have a chance to recover from the initial protein synthesis activity that went on. And so you don't actually add to your memory unless you put those study breaks in there. Okay, so you want to study material, take a break, and study that material again. Repetition with breaks in it improves long-term memory. One other thing, you want to try to get as many pathways to those neurons that have the information as you can, which means you have to use all of your senses. A lot of us as students have a tendency to just read the material, and that's all we do. We read the material, and we read it again, and we read it again. Well, you're just using one pathway then to establish that learning memory. Draw the material out so that you involve a mechanical pathway. Say it. Say the words themselves. Antigenic variation. Hearing yourself say it helps you remember the words, and it creates another pathway to that neuron that holds that information. If you cannot sit there and explain something out loud to yourself, I mean, it's easy to explain stuff in our heads because, oh yeah, I meant to say that. I know what I was saying. No, sit there and explain it to yourself out loud, in the mirror or whatever. If you can't do that, you don't know the material. As soon as you can do that, then you know the material. Just little study tips, study techniques that I've read about in the last couple of weeks. So I thought I'd share those with you. Take it for what you will. Hopefully it'll help some of you. Okay, so we were talking about host microbe interactions. Any questions? How about that term I've just thrown out there, antigenic variation? What does that mean? If I were to use that on a test, would you be able to tell me what, define antigenic variation? Because I said it probably 4 or 5 times in our last lecture about host microbe interactions. One of the ways that microbes are able to avoid our own defenses is through antigenic variation. So what is antigenic variation? How does that help the microbes avoid our immune system? We've got 2 bacteria here, right? for both, let's say they're both E. coli bacteria, and they both have an antigen on the surface. Okay? There's a protein on the surface of these E. coli cells that our immune system recognizes. So this is the antigen. And bacterial cells can have many antigens that our body might be able to recognize, our immune system might be able to recognize. Antigenic variation happens when this antigen changes, such then that we don't recognize it anymore. Okay, so antigenic variation would imply then that the microbe changed the antigen, synthesized a new protein because they have the genes to do so, and now our antibody doesn't recognize the antigen, and so it doesn't bind to it. So when I talk about cells that have antigenic variation, or they use that, we have to understand that means they're changing their surface proteins so that our antibodies don't recognize those proteins. It gives those bacteria a little bit more time to multiply so that they can grow before they get attacked by the other immune cells. Just with regards to the test coming up, know your normal microbiota and what it does for you. We've talked about it twice now. We talked about it in exercise or chapter 15 and here again in chapter 17. You know, what are the functions that the normal microbiota provide? Understand some of the terminology that we threw out there. You know, pathogenicity, virulence, opportunistic infector, infectious dose. And a lot of these terms you've already heard. So again, maybe we're just, you know, repeating stuff that you've heard again. It's going to add to that long-term memory. The postulates that we talked about, host postulates and the molecular postulates. Understand the difference between them and which one we employ today. Okay? Mechanisms of pathogenicity. We talked about 4 different ways that microbes can cause infection. Toxin production, ingestion, then toxin production, invasion of host tissues, then toxin production. Any questions about host-microbe interactions? What we'll see is that it really is going to overlap with material we've already covered and some of the new material that we're going to cover going forward. So today we're going to talk about immunological disorders. I'm a little bit ahead of schedule, but that's okay. These are conditions now where the immune system is not functioning correctly. And the ones that we're going to focus on are hypersensitivities, known as hypersensitivities. And when we talk about hypersensitivity, what we're talking about is your immune response responding to something that doesn't make you sick, but it makes you sick, in a way. Think about hay fever, asthma, hives, those kinds of things. Those are hypersensitivities. The reason that you have those is because your body is responding to an antigen as though it's going to cause damage or that it's something that needs to be destroyed. But in many of the cases, these antigens, in most cases, these antigens that are being responded to wouldn't cause you any ill. We're talking about pollen grains a lot of times, maybe oils and stuff from plants, that might cause this. So a number of different things can cause hypersensitivities, and we're just talking about an immune response to something that wouldn't normally cause you any ill.
Is that in general an allergy?
Allergies, yes, absolutely. A gentleman by the name of Charles Richet along with Paul Portier we're doing some research on the Portuguese man of war, and they noticed that it produced a toxin that was harmful to certain animals, some rabbits and other things that they had experimented on. And then they started looking at the toxin of a sea anemone. And the sea anemone is closely related to the Portuguese man of war. And extracted some toxin from the sea anemone and injected it into a dog, several dogs that is. And many of these dogs survived this initial exposure with no problem whatsoever. Now, Charles Richet and Paul Portier had a background in Antisera, so they had some familiarity with immunology. They took that toxin and they injected it into some of these dogs a couple of weeks later, a second exposure, if you will. And within a half hour, the dogs died. Breathing became rapid. Soon they fell over, diarrhea, vomiting, dead within a half hour. They hypothesized that the initial exposure to the toxin caused the animal's immune response to respond a second time to the point that it led to the death of the organism. It was an overkill as far as its response was. The animals had developed a hypersensitivity to that toxin. A couple of other immunological disorders that we'll talk about, autoimmune diseases and immunodeficiencies, and those will come at the end of the chapter. With hypersensitivities, we're talking about allergies, as Laura mentioned here. There are 4 types of hypersensitivities. Type 1 is known as immediate IgE-mediated hypersensitivity. What? It's pretty self-explanatory once we understand what it's saying here. It is a hypersensitivity that develops rapidly within 30 minutes of exposure to the agent that causes it. It is mediated by the antibody IgE. This is an immunoglobulin E. Remember when we talked about antibodies, we said that there were 5 classes of antibodies? Well, this is one of those classes, IgE, and it is responsible for this type of hypersensitivity. What type of hypersensitivity are we talking about here? This is the hay fever, the allergy, asthma, that kind of hypersensitivity. I'm sorry, was there a question?
Was bee sting would be included in that?
Yes, actually, a bee sting would fall into this, and we'll get into that here shortly. Now, one of the terms that Charles Richet and Paul Portier coined, having observed the response that these dogs exhibited on second exposure, was the term anaphylaxis. Actually, I spelled this correctly. And it was just the opposite of prophylaxis, which was a protective measure. So the immune system was prophylactic in that it protected us, except for in the cases of hypersensitivity where it was anaphylactic. So we hear this often, we hear the term anaphylactic shock. These hypersensitivities that are immediate are referred to as being anaphylactic conditions. Okay. There are several types so we can talk about, but before we get into that, how does this hypersensitivity develop? Well, you get exposed to the antigen. And when you are exposed to the antigen, that means then that the macrophages and or B cells take up the antigen, right? And they're going to present this antigen then to a T cell. We find that in our mucous membranes, lining the tissues of our mucous membranes, high concentrations of B cells. So now, the macrophage, the B cell, the other one, presents the antigen to the T cell, and the T cell then activates the B cell. Just as we would expect from any normal antigen exposure. Of course, we have to understand that this is an effector T cell, meaning that it's been presented the antigen by a dendritic cell at some point prior to coming in contact